Go. And we are now recording, just reminding you that this is just a visual on the screen, which is basically what we're going to be looking at here. And it's recording our voices, okay? So the mic is not that sensitive, so if you actually do have interaction and we talk about things, if you would try to kind of speak up for Luke to be able to hear what we're talking about, okay? Oh, hang on. I'm sorry, this is family. I never get a call. Hey. Hey there. Hi. How are you? Are you alive? That's right. It's right there. I did. Sorry. No emergency. My heart usually leaps up into my throat when I get a call in the middle of a working day from family. Okay. And we got Andrew here. Got it. All righty. So as my This Week in Plant Ecology beginning of the semester, I want to go ahead and dive into talking about our lecture material. We are talking about plants and their environment, okay? The ecology portion of the plant ecology title means that while we know some basic botanical truths about how plants work, there's more to know about those things, about the way that they tweak the response to the environment, or the way that they adapt to limitations or excesses in the environment, okay? So one of the things we do is we remember some of the basic botany that we know, and this chapter starts with light and photosynthesis, because what could be more essential for the well-being of plants? In fact, if you'll notice, we've got plants over on the table there, and they're in front of a window because they need light. And if they are starved for light, they are not going to thrive. They're not even going to survive. So interestingly, though, when a plant is deprived of light, there is a way that they survive for a short time, and then upon exposure to light, they respond very quickly to recuperating. So one of the things to think about is what happens when you don't get something, and that's, again, plant ecology. Alrighty, so in Unit 1, which we're looking at literally the individual and its environment, we're going to be talking about photosynthesis in the light environment. And generally speaking, I don't have an automatic thing here, so I have to come over here and kind of thumb down here for you to see. This is posted in Moodle, so you can pull it up and take a look at it there. We have a chapter outline, and then we get into looking at some of the information. I often bring in diagrams and pictures and other bits of information that expand on or improve our discussion of certain bullet points. So while you're going to still see things that are important out of the chapter, you're going to also see some reminders. Do you remember looking at this in botany kind of thing? So, okay. So those are the kinds of things that we're going to actually see as we unfold our discussion. Also, along the way, you're going to see activities that we're going to be doing, and you're going to need your textbook. So, coming? Still coming. Okay. I was supposed to be here yesterday, but so hopefully today. All right. Keep on top of it, and if not, remember the bookstore has them. So you need to have that book by the end of this week, or we're just treading water, and I don't want you to sink, okay? So make sure you get what you need. Alrighty, cuties here for anybody who would like a really yummy, pass them around. So as you can see, we're going to have some opportunities for doing in-class activities. And you're going to need your textbook, and you're going to work in small activity groups to work through some stuff as well, okay? I like to use the textbook. You're paying money for it, we're going to use it. But I do bring things in, including literature, and I bring things from examples from other places as well to kind of augment what we're talking about in the topics. Make sense? That's how kind of things will unfold in the class. So backing up, let's see here. A couple of things to think about when we talk about plants and the environment, photosynthesis in the light environment. Plants go about individualized, unique ways to acquire resources, to acquire energy, and to acquire materials. We acquire materials, we do that by eating and drinking, don't we? Because we have to take in resources, and the only way that we do that is by ingestion, right? Well, plants make their own food. We eat other things, but plants make their own food, so they have a unique way of acquiring energy, okay? But they still need to bring in carbon to make carbon-rich molecules. They need to bring in elements that are necessary for membrane activity and just the health of the cell that's in the plant, so they need to acquire nutrients and other resources. So acquiring energy and materials is, again, something you could spend a year. People have spent their lifetimes understanding different ways in different environments and how plants go about acquiring energy and nutrients. So lots of papers out there on that one. Limiting losses. That means you're going to maximize your efficiency. And we learned a lot about how photosynthesis works in plants and the enzymes that are involved. And we know something about how plants can optimize their photosynthetic rates, how fast they can photosynthesize, how well they use certain wavelengths of energy. Okay, so we can study that in greater detail as well. So limiting loss, such as opening your stomates to let carbon dioxide in, but when that stomate's open, water's getting out. Okay, so you have to go about limiting your loss, and one way to do that is to become more efficient at making your own energy, more efficient at photosynthesis. So again, you're going to see this in just a minute. C3 pathway, C4 pathway, CAM pathway, and how those limit losses. Okay. Another one is being able to allocate resources to basically move things around, to move resources around to maximize strategies within the plant to reproduce, make more of themselves, or simply to just stay around from day to day to survive. And depending on the environment that we find them in, you're going to see different types of plants might, and I say this in an anthropomorphic kind of way. Plants decide that they need to move things perhaps to a stronger root base because water's limiting and we need more root mass in order to pull up more water. So allocation to a better underground system. Okay. Another one is, this is fine, but we're in the shade and we need to capture as much light, so we allocate resources to surface area to be able to collect more light. So moving things around, moving resources around to maximize how we interact with our world, allocation of resources to maximize survival, to maximize opportunities to reproduce. To capture and incorporate by photosynthesis. PS is photosynthesis. Oxygen, carbon dioxide, water, and light. Now, we already know that plants need CO2, and CO2 is useful during the process of photosynthesis, right? To build carbon molecules, exactly. But remember that these guys also do cellular respiration, aerobic cellular respiration. So we still need to have oxygen. Plants don't survive long in an anaerobic environment. So we do need to have oxygen, but oxygen, as we're going to recall in just a couple of minutes, is a competitor for how enzymes work in photosynthesis. So trading those things off. So we talked a little bit about C3, C4, and CAM in botany, but now you're going to see why it's kind of essential for us to understand in terms of plant ecology, right? Alrighty. Adaptations we might be focusing on under the photosynthesis and light environment thing here is adaptations to limitations of light and to limitations of water. We're going to see that unfold in our chapter. And adaptations to optimize uptake and use of nutrients, being able to get as many of those resources as is available. And when we talk about optimization and adaptations, we're really getting ourselves set up to start talking about competition, right? So if you're just, here you are out there and there's nothing around, you're on the Mars landscape, what you do is all about just you being able to survive, right? But if you're on South American terrain, tropical jungle, and you're surrounded by other organisms that often need much the same that you need, and the resources that are out there are limited, now you're talking about how well you do things and how well you survive depending on how well you compete with this guy over here. Okay, so competition is an essential component of discussion of ecology. So it's an aspect of ecology we'll be talking about. Okay, so far, 5, 1, 2, 3, 4, basically 5 different things that we've bullet listed here just to kind of remind you about plants. Okay, lots of theory, lots of studies. Why does this thing keep doing this? Sorry, Luke. Things are popping up on here and covering up the screen as we're recording. What's going on the screen? Stop it. So, okay. So when we talk about the individual and its environment in this unit, this chapter, depending on looking more closely at photosynthesis, photosynthetic processes, adaptations, optimization, all that kind of thing, we need to talk about the physical conditions that the individual experiences in its environment. So physical conditions affecting plants. Okay? So features of the environment that can affect the physical condition of the plant, such as precipitation, water. Very good. The obvious one is light, so let's give that because we've been talking about light and photosynthesis. Precipitation, and we kind of had that on our board just a little while ago when we were talking about what could we do for our study, right? What could we look at? So such as precipitation or water availability. What else? Soil. That could be
nutrient content as well.
And what else about soil?
The amount.
Okay, that would be important. Sometimes plants are not rooted in soil, right? So they have a very unique way of adapting to their environment. But also, just the very character, the very nature of the soil is important. Knowing how much organic matter is in there. Knowing the texture. Are they large boulders versus small bits of clay? Okay, one of our lab activities is going to be characterizing soil. So you guys can actually see, yeah, we can measure this. We can evaluate this. Okay, so yeah, soil is good. Anything else? Features, we're focusing here on features of the environment, such as things that physical conditions that affect plants. Anything else that you can?
Other plants.
Other plants. Yeah, exactly. In addition to the physical, we also have the biotic components that would make up the features of the environment. However, if we're sticking with physical conditions, we're sticking with abiotic components, right? Make sure you see the distinction between the living components of the environment and the non-living. So again, water, sunlight, along with sunlight temperature, right? Very cold, very hot, could be very stressful. Very cold, very hot, very stressful. Water, did I already say that? Nutrients, soil type.
Did you say space?
Space. That's a really good one. Just how much area do you have for growth and reproduction? Space becomes an aspect of competition, though, but it is the physical factor that plays into us talking about the biotic component of competitions. That's a good one, especially for plants. So we're looking at density, right? Space translates into something we will talk about in terms of density. You have an area, how many individuals are in there, okay? So if we have an area, a box, compared to another box, because you have to have something to compare it to see the difference. If we have one box with individuals with 1, 2, 3, 4, 5, 6, 7, 8 dots in it versus another box, let's say we've got 50 dots in it, individuals in it. Which of those two, the box on the left or the right, has greater density? Right, and therefore space is the issue. So we're literally, when we say space, in terms of plant ecology, we're looking at density of plants. And that's something we can measure. We can actually go out into the field, set up a survey technique, a method, and count numbers of plants in a given area. So we can actually evaluate density of certain plants. And we can evaluate that in terms of competition or limitation of resources. There are lots of different ways that we can look at space. A great place to start. One of my favorites. Okay, so then getting into the other living organisms, we're looking at competition. Exactly. So just other critters. Not only just other plants, but other living organisms are also predators, such as herbivores, right? Not just grazers, herbivores, but we also have parasites and viruses that attack plants as well. Very important if you're looking at an agricultural slant on understanding plant ecology, because plant viruses and resistance to plant viruses are often the focus of a lot of research and agricultural research, being able to resist large monocultures, a field of corn, one plant, large area of it, a one culture field of plants. They're going to be very susceptible to diseases and viruses and things like that. So we genetically breed certain strains of plants that may have inherent resistance to certain types of viruses and pathogens and things like that. Not a lot of resistance to fungi, as far as I know, can be bred into a plant, but you do treatments and things like that that can help with those things. So other living organisms, plants beside plants, but also plants beside animals, that can be a good thing too, right? Because if you've got plants sitting next to some animal that doesn't eat plants, maybe the animal excretes or releases nutrients into the environment near the plants where they're habitating, it can be a good thing for the plants without them suffering due to, I mean, there's lots of scenarios I can think of in my mind where you can talk about stuff like that, right? Good and bad stuff. So, living organisms. For example, ecologists study how plants respond to all this and more. And yes, there it is. So light availability as affected by competition. If you are in the understory and you're shaded underneath larger plants or larger leafed plants that are blocking the sun, then you are directly competing. What you might notice then is the plants that are growing in the understory might have a growth spurt, and they shoot up in order to escape that limitation of the understory. So you can do studies to evaluate how plants respond, where plants respond in their body structure, as well as how well and how fast they respond to those kinds of limitations. And lack of light is a very great stimulus for spurt of growth, to get out from underneath that limitation. So light availability affects competition for plant light. Also, light availability can be affected by pathogens. The healthier you are, the more resistant you are. So the more sickly you are or limited you are, the more likely you'll succumb to pathogens. It makes sense. We experience the same thing. Pollutants, same thing, herbivory. There are lots of strategy that plants employ to reduce their obviousness. I guess you could think about it. One of the ones I love to talk about is the sensitive mimosa. The plant that I'll try to get some seeds and we'll grow some, because I haven't grown any of those in a long time. But the plant, once it is in a little plantlet stage, if you touch it, let's say it's out like this with its leaves, and if you touch it, it will fold in its leaves and then fold down, becoming very small, reducing its surface area, so herbivores won't see it. And deer just walking through the forest, disturbing the vegetation, that sensitivity will cause it to close up, and then it has a greater possibility of not being eaten when you can't see it. So lots of different strategies to resist herbivore. That one's a sensation one and an avoidance one, but there's also toxins, and there's also you can eat me but not get a whole lot of nourishment out of me. And herbivory can actually spark a communications network between stands of plants. But there's an herbivore in our midst, start making anti-herbivory compounds that will ward off being eaten. So there's even a communication pathway amongst plants about herbivory. So that one has a lot of different things that ecologists study. So that's just a basic overview of looking at the things we might consider, and kind of jumping around a little bit, but thinking about the breadth of things that we can think about when we just think about simple things about plants, okay? So let's take a look at the chapter outline, and we'll go from there. We'll start into our content in the chapter. We'll probably get up to, we'll see how we do with getting up to that comparison table. I might make that an assignment that you can then bring back into class, and we can work on it together after you work on it at 11. So chapter outline, class time will be spent on these topics. Lab time will be spent researching this topic. So for instance, we'll be working on soils, soil composition, soil organics, soil moisture, and so I'm going to be doing that to demonstrate methods to you. Remember, this is a field methods rich course, so we're going to be working on that. Okay, so looking at chapter outline. We're going to start with the process of photosynthesis, a little bit of review. Then we're going to talk about how ecologists can evaluate, compare, and assess photosynthetic rates, looking at differences in light levels, carbon dioxide uptake, and with that differences within and between habitats. How are the rates here versus there, and why? Why are they different? If you're comparing the same species over here to over here, and yet you measure, and you can measure their photosynthetic rate, why are they different? How are they responding differently? What is affecting their photosynthetic response to their environment? And that's an interesting question to look at. Okay? Again, we're going to look at our three pathways, a little bit more detail after a quick review. C3, C4, and CAN. And we'll talk about evolution of the three pathways. We'll look at phylogeny and how they developed through evolutionary time. We're going to look at form, phenology, which is basically just a timing aspect of the way things unfold in the life of a plant. Seasonality, if you want to think about it that way. and just distribution, how those things interact. So we'll look at patterns of distribution and abundance, where we find these three different mechanisms of photosynthesis. We'll look at adaptations to light, sun plants versus shade plants. If you remember from photosynthesis, or from botany, when we studied photosynthesis, I showed you some microscope slides looking at the differences in structure of a sun plant versus a shade plant. So we'll revisit a little bit of that. High and low light habitats and day length responses. Things that we'll be paying attention to. Now, first thing I want to do before we get into this is just we're studying light, okay? And I want to give you some perspective, maybe a review, maybe a reminder, maybe new information on light and what light is as an energy component, as a physical nature of light, because that's important for us to understand how plants use the dynamics of light. Okay, so we're going to start very simply. I'm going to talk a little bit more about light. Okay. Okay. So light has two features. I'm trying to think of a generic term to discuss light. There, we can measure light quantity, if you will, intensity. And you might use words like highlight, low light, shade light, bright light, right? Those kinds of things, thinking about light intensity. But you can also measure the quality of light. And that, I mean, that word quality connotates something very unique, right? It means that there is value or lack of value in certain aspects by measuring quality of light. When we break light down, we're going to come back to this, we'll relate it back to here. Light can be described in terms of wavelength. Units are in nanometers. And it's literally the length of the wave as we measure the distance from peak to peak. And if you have a continuous scale of distances from very close to each other to very far apart from each other, we can lay that all out onto an axis and call it a spectrum of light. And we can have wavelengths with peaks very close to each other, and we can have wavelengths very far apart from each other. So if we apply a descriptive characteristics to these differences, we're looking at high energy wavelengths where the peaks are very close together, and low energy wavelengths where the peaks are farther apart. I've always talked about this in terms of, have you ever stood on the shore of a lake or the ocean, mostly a lake, when a storm is brewing and the waves are really choppy, and as they come into shore, they're very frequent. Slap, 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 slap. You're standing, thigh high in the waves, right? So high energy is when slap, 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 the peak hits you very frequently, and you can feel that energy. On a nice calm day, you might be standing thigh deep in the water and there's very few waves coming to shore. So there's an undulation and you feel a little bit of a wave, and then a long time goes by and then another one might come by. So it's very peaceful, calm, and there's no agitation and energy involved there, very little. So the energy on a spectrum of light can range from very close, very small wavelengths to very large wavelengths, kilometers, literally kilometers distance between peaks, to even smaller measurements of nanometers. And then we can overlay spectral colors on this scale, and we're running from ultraviolet and blue all the way up through green, yellow, orange, all the way up to red and far wavelengths of light. We already know, you know where I'm going with this. We know that when we study photosynthesis, that only certain colors of light are extremely important in the actions of photosynthesis in plants. So we know that blue light is very useful, and we know that red light is very useful. We know that green light, because green is reflected off of plants, green is not being used by plants, so it's not a very useful light color. However, other pigments might pick up something like yellow or orange light and use that as accessory lights and accessory pigments. Okay? So light can be thought of in terms of the distance between the waves, wavelength. Another way to think about light is its intensity as measured by photons, which is a physical component. A photon is a discrete unit of energy that we measure light by, the intensity of light, photons. And I like to think about it in this, it makes sense to me to think about it in this way. We have wavelengths of light that are traveling from source to what is being hit by it, matter that's receiving this energy. And traveling along this wavelength, we have these discrete packets of energy. Now, if the packets of energy look like this, for example, and then another one might look like this, photons of energy traveling along that wave pattern, which one, top one or bottom one, has higher intensity of energy? Let me back up. A photon is a discrete unit of energy, a physical packet of energy that we can see and measure, okay? And they travel along these pathways of waves, okay, the paths of waves. So if we're hitting material or matter or pigment over here, is this thing seeing high intensity, low intensity, comparing these two things? Which one is high?
The top one?
Yep, top one, because more photons traveling along that pathway. Making sense so far? Okay. Now, here's something to realize, that we can actually measure this because we have been able to construct a material that can measure how many times it's pinged by a photon of energy. And we call a photodiode array. So when we construct sensors, we make it of this material that when light and the photons traveling along the wavelength of light hit the sensor, we can say 1 photon, 2 photons, 3 photons. You know, in some of the older instruments, you actually hear it clicking each time the photon hits. That's the way we used to measure listening. How many times do you hear little ticks when the photon hits the sensor? Now we've got data loggers that do the math for us, that listens for us and then gives you an output. Well, I saw 50 photons hit the sensor in this amount of time, okay? So we can actually measure light intensity in that way. Other sensors pay attention also, or more attention to the actual distance between the wavelengths to tell us what color of light we're actually seeing. Okay? So light has a pattern and it has a unit. There's a physical component to it, the photons, right? Now, this material, man-made material, can be the photodiode array that we see in the sensors that our instruments can be used to pick up this stuff, or it can be a pigment. And that pigment is receiving photons of energy. And when that photon of energy hits the pigment, it excites an electron up to a higher energy state. The excited electron gets handed across the membrane, giving up a little bit of energy along the way, going from a higher to a lower status of energy absorbance. That energy gets used to do work. And then we hit it again with more photons of energy, excite the electron to a higher energy state, and it goes back down across the membrane again. We call this the Z scheme. Remember that? Yeah. Way back to that stuff you thought you'd never have to think about again. So light, we can measure quantity or intensity. We can actually measure this, and plants can use this. And we can actually see how plants respond to differences in intensity and wavelengths by measuring the output, the payoff at the end of the day when the pigment receives X or Y. Does it grow? Does it make more chlorophyll? Does it reproduce better than another set of plants who gets a different set of wavelength and intensity? Lots of endpoints that we can study, measure, and see if there's actually any difference between this light environment and that light environment as it affects the plants that are being exposed. Okay? All right. So light. We can also measure quality of light. One of the things that I did for my dissertation research was a question I found fascinating. There is a particular type of pigment in plants called phytochrome. And it is literally just like this. Phytochrome has an on switch, and phytochrome has an off switch. When it's on, it's physiologically active. When it's off, it is not. So then you learn, well, what turns it on? Why is it off? And when it's on and physiologically active, how does it affect the plant? What does it make the plant do? And if it's off, what does it make the plant do or not allow the plant to do? Keep it from receiving what it needs. Okay. When I studied phytochrome, just from a very generic standpoint, phytochrome is affected by ratios of red to far red light. And red and far red light is affected in forests when sunlight shines down through a canopy. The leaves in the canopy preferentially absorb red light, which means that the red light above the canopy and the red light below the canopy changes because the light is passed through the upper story canopy. Okay? Oddly enough, there's also far red light, which, remember our spectrum, there's UV, blue, green, yellow, orange, red, and then far red, right, off to this end. So we're actually talking about out here on the long wavelengths. And the far red light in solar radiation pretty much is not affected. It passes right on through. So really, we've talked about a ratio of far red red to far red above before the canopy and a ratio of red to far red below after the canopy, and how it affects these guys living in the understory and how phytochrome on off switch works. I'm being very vague because we'll get into a little more about that later when we talk about that. But this switch on and off reads the environment. It tells the plant what's going on in the world around it. Believe it or not, plants can sense what their environment is showing them. So, how does a low red far red ratio affect an understory population of plants that are limited by light? We believe that depending on the changes in the environment, it can stimulate that plant to grow, to elongate, to shoot up perhaps into a greater light environment. Just depends on where it's at and what kind of duration it receives to different or varying red to far red ratios. Bless you. Phytochrome on, phytochrome off. On, off. On is like this. See if you can tell me what this is. An hourglass? Okay. Yep, kind of works for you. Basically, we turn this thing over. All the sand is in the upper level, and it works its way down through the hourglass, right? Okay. So when we hit phytochrome with light and shift it into its active form, we're basically shoving all the sand into the upper chamber of the hourglass. We've switched the hourglass. And then slowly over time, as the sand slips down through the hourglass, we're going to see a degradation, a movement from active to inactive form of the molecule. And the amount of active to inactive form of the molecule can determine how enzymes interact, how enzymes are made, how chlorophyll is produced. This is a really big one for plants to make more chlorophyll, and we know that that means you can get more light energy, right? So phytochrome reading its environment and acting kind of like a timepiece is fascinating to look at how plants adapt to their understory environment. I took this question to one of the most unique environments in the world, to a montane cloud forest in the Amazon, the Andes part of Venezuela. And the clouds, the mountains were so high that the clouds literally grazed the top of these mountains all day long. It was just like the peaks were just jutting up into the cloud canopy, the cloud overcast. And we had tall trees and we had plants underneath that. So in addition to being shaded by the overstory, these guys were like virtually in darkness because of the lack of light getting through with the cloud cover. So montane cloud forest proved to be an extremely interesting place to answer, to look at and study this question. How do these guys manage to survive, and what is the light environment like underneath these areas? So we took sensors that could measure red and far red light, and we surveyed the understory areas and then related it back to where we saw plants and where we didn't see plants as part of our study. So interesting stuff. As far as I know, I still have that unit, so I have that equipment that we could, if you wanted to look at colors of light, it might be something we could address if you were interested in asking a question along those lines. So, light. Photosynthesis, yes, but also, look, activity, turning enzymes on and off, acting as timepieces, being able to read the environment and changes in the environment. Some interesting stuff going on with light and plants. Okay. Okay, so high-low light habitats, and last one on our list there is day length responses. This one's really important when you think about phenology. Again, that timing thing that I talked about, phenology. And we see seasonal changes in the way plants do business. Why, do you wonder? Of course you wonder. Why do plants not bloom, set seed, and make babies and send them off in the dead of winter? Absolutely. Temperature and sunlight available. More particularly about light. She said sunlight. We've got day length, night length. Okay. Phytochrome plays a role in this too. But what we have in the winter are long nights, right? It gets dark early and it doesn't get light until really late in the morning. Long nights. Turns out it's not the actual day length, but it's actually the night length, because as soon as you hit phytochrome with light, you reset its little time clock. So the longer you don't interrupt the phytochrome activity, longer nights, then plants are not motivated, not excited to, not set physiologically on the path of waking up, making leaves, growing roots, making flowers, setting seed, making babies. Okay. But in the spring and summer, day gets longer, night gets shorter. And so you've got more frequent interruptions of the long night times. Phytochrome turns on to its active form, plants start to leaf out, make flowers, set seed, etc. So literally one of the predominant factors in the environment is light, but it's its absence and the interaction of light with phytochrome that seems to make the most difference. Okay, so we've been talking for a while here, okay, about light and about things that I think might be new to you, and also reminders of things that we've talked about before. And let me say that what I would expect you to be able to do if I asked you a question on the exam, for example, one of my questions could be, we talked at length about several things to know about phytochrome. Could you give back to me an understanding of what we just talked about with phytochrome? I didn't get into the particulars, but at least an initial understanding you should be able to relate that. Also, light. If I ask you to describe light to me, can you do that? Can you talk about light intensity and light quality, quantity and quality, right? And then give me an essay of your understanding of what we've talked about thus far. Now, all of this has been dependent on lecture. You've heard me do this, and I've written some stuff on the board for you to take note of. But this information that I've shared with you is not in the book. Not yet. So what I want you to know is that I will ask you questions over lecture content. So if you don't understand, you need to ask. And take some time to go back over it after we're done with lecture and see if it all makes sense, and you could actually write a little story about what we've talked about, answer in an essay format. Any questions about the generics of that? Okay, that makes sense? Okay, just keeping you on your toes and letting you see where we're heading and how we're doing things. Okay? Right. Let's see then. Good. Then what I want to do is get into our chapter. So generically, plant ecology is ecology applied to the study of plants. So even if you were in an ecology course, I would tell you that of primary importance as an ecologist, you study patterns and processes in order to understand how things work. or don't work in certain circumstances. Okay? So you observe patterns. Pattern observation is something very important to being a successful student of ecology. In fact, you've heard me talk about this more than once. Turn on your observational eyeballs. Look at the world around you. It's a silly exercise to ask you to think about this, but hey, as you're walking across to go to the cafeteria and you're looking around going, oh, look at that. I wonder why it does that. Oh, I know why it does that, because I learned something in another class to help me understand a little bit more about that. So you're really using it very consciously, what you're learning, and you're being very observant about the world around you. It helps you to generate good questions and kind of narrow down what you want to do with your life when you grow up, kind of thing. Okay, what are your passions? What are you interested about doing? Do you like research? It doesn't have to be plants, but do you like the inquiry process of doing science? Then maybe research is something you can think about doing in your respective disciplines. If you like being your own master, you might think about the difference between earning a master's and a PhD. PhDs set the agenda. Master's students more or less work on the agenda. Does that make sense? So PhDs are more in charge of setting the agenda. Master's level education is more about being equipped and experienced to execute the agenda. That way. In some cases, it's a little more, a little less. I'm being very generic here. Okay. Doing general science graduate programs, you can find programs that will pay you to go to school. I think Luke just told me he got a job going to school at a position going to school at Kennesaw, and he's going to earn not a shabby income for being a graduate student and working on a project while he's getting his degree going to school at Kennesaw. So this is something that is different than going to a professional program, where generally you pay them to get your certification and your degree. And it can get expensive. You just need to keep your eye on the ball and are you going to be able to get a job once you're done, that kind of thing. So things to think about as we go through this. But observing patterns and studying processes is just kind of the whole ball of wax when it comes to ecology. So in Unit 1, we're going to be focusing on functional ecology, also known as physiological ecology. What's the physical response of an organism to the effect of its environment? Does it make more chlorophyll? Does it turn on or off enzymes? Does it grow? Does it reproduce? Does it not reproduce? What is it doing physiologically? Okay? Does it kick up its photosynthetic rate or slow it down? That kind of thing. Okay, so just a very brief review of photosynthesis. Just briefly described for you the Z scheme a minute ago, where on the left here we've got pigments. They get hit by light energy, photons of energy, excites an electron up to a membrane acceptor, membrane-bound acceptor, and then it comes back down from low to high energy. It comes back down to a lower energy standing. As it does that, it releases energy for work, and in that work, we produce ATP by phosphorylating ADP. Okay, we do that by pumping hydrogen across the membrane. Remember chemiosmosis? The work is doing the pumping hydrogen across the membrane, and then allowing the hydrogen to zip back through the membrane is where ATP gets made. So it's kind of like letting water flow downhill. But you got to get the water uphill first, and that's where the work comes in. Okay? So then the electrons that are traveling along this Z scheme come to rest in a photosystem here. They get hit with more light energy, get excited back up to a membrane-bound acceptor, and in the process of running from higher to lower energy status, that energy that's released is used to make ADP, H. in photosynthesis. So hydrogens come to rest in ADP, electrons come to rest in ADP, producing NADPH. This is a little bucket of electrons with a little bit more resonance energy still residing in that excited electron. It hasn't all come all the way back down to its resting state. It still has a little bit of work to do left in there. So that's why we can take NADP and move it over here to the light independent reactions and use that energy to do more work. So that comes over here, as well as the chemical energy in the terminal phosphate that ends up on ATP. We bring ATP over to the light independent reactions and get more work and more energy investment over here. Makes sense so far? Remember the Z scheme and all that? Okay, and then over here, this is all about 3 stages of the light independent cycle. First thing that happens, let's get you guys talking and see how you do. So if we were to kind of break this up in this, we'll call this 1, 2, and 3. Do you guys remember this? Maybe, hopefully. You've been a little longer for you than these guys since we've talked about it, but what do you think? What's the first thing that happens in the light independent reactions?
Carbon fixation.
Bless you, child. CO2 fixation. Exactly so. We're bringing carbon dioxide in an inorganic molecule and we are fixing it into an organic molecule. It is the only it is one of the most important mechanisms we have for bringing an inorganic building block into the living world. Bacteria can do this, algae can do this, but plants and all the chlorophyll in the tissues that make up the green leaves of plants are probably in direct competition with all the algae and bacteria that are in the oceans. I've seen textbooks say that carbon fixation going on in aquatic systems is predominant over what's going on in terrestrial systems. I don't know, because I also hear in reading and studying that chlorophyll in terrestrial systems outmatches what we see in this minuscule surface layer of oceans and lakes. So I don't know. Be an interesting thing to find out more detail on where we are in studying it these days. But photosynthesis starts the whole band, getting the band on wagon, right? Starts the whole process. CO2 fixation. Next thing that happens, anybody? Let's think about it this way. This one has to do with, these steps that are in this region here have to do with carrying NADPH, which is a little electron bucket, and bringing them in and dumping electrons into the compounds that are cycling through the light independent reactions. What are we doing with the electrons? Are we taking electrons away from these carbon-rich molecules and handing it to NADPH? Or are we taking the electrons in NADPH and handing them to the molecules that are in the cycle? Was I clear in my question?
You're taking them from NADPH and putting them in the cycle.
Thank you. Okay, so when I have an electron, you guys remember this, right? When I have an electron, Catherine, and I hand you an electron, I've been oxidized and she's been reduced. Thank you very much. So when electrons get handed from NADPH to the carbon-rich molecules in this cycle, what are we doing to the carbon-rich molecules? Oxidizing them or reducing them? Thank you. So what's step 2?
Reduction.
It's reduction of PGA, right? The first 3 carbon molecule in the system. It's reduction. We're handing electron energy over to that 3 carbon molecule. Yes, and this is a cycle, right? Step 1, 2, 3, and we regenerate the initial molecule, right? We always come back to the dog chasing its tail, where the teeth meet the tail. And step 3 in a cycle is always about regeneration of the initial molecule. Always. Or it's not a cycle. Does that make sense? Remember this stuff? How many of you are in biochemistry at the same time? So this is going to be, yeah, you're going to get this. Hopefully you've already got it, but you're going to get this. Okay? So we've made ATP, we've made NADPH, and what else have we made? What is this? Oxygen. We did that by doing what? We split water. Or rather, the plant does. We don't. The plant does. The plant splits water, known as photolysis. It uses light energy to split a water molecule, frees hydrogens, frees electrons, frees oxygen, so that when we look at the overall equation, CO2 plus H2O, there's our water, gives us O2, there's our oxygen, plus C6H12O6. And that's our sugar that spins off the cycle to go through sugar synthesis process. Following along, see that? So there's our equation. I don't care about it being balanced. We bring CO2 in here. We split water there, and we do that so that we can replenish the missing electrons here that disappeared over here. So if the electron ends up over here in NADPH, we got to give it back to the photosystem here so that we can actually continue the process. Otherwise, we're going to run out of gas. So how do we replenish it? We split water, release hydrogen and electrons, and they get replenished here so that the next bolt of light energy can excite the electrons to a higher energy state, and so on and so forth. That's the mechanics, that's the physiology, that's the basics of photosynthesis as we know it. PGA is a 3 carbon molecule, so we call this pathway the C3 pathway. The enzyme that is critical to this process, to the activity of fixing carbon dioxide into the initial molecule, or into the first product of the reactions. What's that enzyme called? Do you know?
Ribose.
Yeah. Ribulose rubisco. Ribulose bisphosphate, or rubisco. And we know something interesting about that enzyme. It's probably the most abundant enzyme on planet Earth, first of all. But it also has an evolutionary capability of interacting both with carbon dioxide to do this carbon fixation thing, but it also will interact in its active site with oxygen, making photorespiration a process alternative for rubisco. Now, in some circumstances, that can be good for the plant to be able to do photorespiration, but largely it's a very inefficient way of fixing energy through photosynthesis, because you have to go back and reclaim all the molecules you've split apart and gone through respiration with the process of photosynthesis. So we we think of it in terms of when is it good for the plant, when is it not good for the plant, what environment, what circumstances, what situation, and its adaptability, how it affects its adaptability. So, when plants photorespire, when rubisco gets together with oxygen instead of carbon dioxide, what happens? And we have photorespiration, the three pathways of photosynthesis. So the plant is in a hot, arid environment, and it has to bring in carbon dioxide to do fixation. So the leaves open the stomates. CO2 has to come in. But it's hot, dry, windy, and that means water inside the leaf of the plant leaves as soon as that stomate's open. Now, depending on the resources in the soil, can I replenish that water into the tissues of the plant so I can continue to split water molecules, free electrons, and keep the process of photosynthesis going, right? Okay, sometimes photosynthesis gets stalled out by photorespiration, essentially more vulnerable in hot, dry, windy environments, arid, dry, windy environments. So what we have here is a process. Let's see if I can bring that down just a little bit. Photorespiration. First of all, let me point out that it happens in the leaf cells, the chloroplast of the organelle, chloroplast organelle in the leaf cells. But in order to reclaim the mistake, it has to go through a cleanup organelle called the peroxisome, which is just like hydrogen peroxide. It breaks things down, right? And it also has to go back through the mitochondria. So it happens, it gets sent through the peroxisome down to the mitochondria, this way, sorry, down this way, and then it comes back up through the peroxisome, back up here, and gets made into PGA so it can go back into photosynthesis, back into the light-independent cycle where PGA is. Look at all that, just to get back to PGA, okay? So what we see here is in the chloroplast, we have glycolate, which gets transported into the peroxisome, becomes glyoxylate, glycine. Glycine gets transported into the mitochondria. We use oxygen, reduce it to decarboxylate glycine to serine. Serine then can get transported back into the peroxisome where we hand it some electrons, right? And then it gets transported to, transformed and transported back into the chloroplast as glycerate. It gets phosphorylated. We donate a phosphate from ATP. Glycerate, 3-carbon, becomes 3-PGA. Glyceraldehyde, GA is glyceraldehyde, glycerate, glyceraldehyde. Add a phosphate from ATP, and now you've got PGA. All of that to just get the 3-carbon molecule necessary to plug back in to right here, the PGA right here. So right where the enzyme not really malfunctions, I don't like to say it that way. It interacts with oxygen instead of carbon dioxide. It goes through that entire process just to regenerate back to PGA so that it can continue on around the cycle. Very energy inefficient, lots of organelles involved in the process. You can imagine just how much energy that takes to do that, especially when you're using energy ATP and NADH in order to make all this happen. So here off to the side is a discussion about what happens with photorespiration. Use of oxygen and RUBP. RUBP use can be formed by photosynthesis only. So making RUBP is our first molecule, right? RUBP is the initial molecule in the cycle. Remember that, 5-carbon molecule. Now, RUBP and Rubisco and oxygen interact, and then we end up with the function of photorespiration, and we need to recover some of the carbons from the excess glycolate. So glycolate gets transported into the peroxisome, transformed to glycine, imported into the mitochondria. Oxygen is used and carbon dioxide is produced. We decarboxylate glycine to serine. Serine then gets transported back up. We hand electrons to serine. We hand phosphates to serine to make glyceraldehyde into PGA. All of that to recuperate what we lost simply because at the start of the cycle, instead of carbon dioxide, oxygen and the 5-carbon molecule interacted with Rubisco. That's photorespiration. It's called respiration because this is what's happened. We have peeled off a carbon, respired a carbon. That's why it's called photorespiration. All of this due to light activity, hence photo, respiration in the mitochondria, hence respiration, photorespiration. Okay? So that gets us to photorespiration. And when we get back together, we're going to talk about C4 and CAM pathway and why plant ecologists are interested in knowing more about these processes and what's important for us to understand for the success of plants in their respective environments. Remember, I talked about an environment being hot and windy, right? That's one such environment where we need to see this working. Good to go? All right, Thursday, class, picking up where we left off, and lab, getting together and trying to nail down what it is we want to do for our study. I need to order stuff, so we need to kind of get busy on defining what we want to do. All right? All right, guys, see you later.